So, yesterday we looked at all of the events that lead up to a cell preparing for division. So we went through all of the growth that's necessary, the division of genetic material that's necessary, and the accumulation of goods that are necessary. And I think I alluded to it in here, and I know I elucidated it for the other hours, where I said G1 is like, for a cell, it's the same process as from when you were conceived as a child all the way up until puberty. S is puberty. It's where all of the necessary things happen inside of the cell to prepare for division. But, like I emphasized to 7th hour, and I feel like it's my public duty to do for you guys as well, just because you're physically capable of reproducing doesn't mean you should. Isn't that right? Excellent. Glad I can make sure I emphasize that for you as many times as I can.
I like the way you put it last semester.
What?
No, last semester you told us that girls had cooties and showed the girls that guys had cooties.
Yes.
I
Well, now half of you have been through health now, so I can't lie about this cooties thing anymore.
What? Girls don't have cooties? What?
No, we still do. Stay away from us.
Oh, okay.
But I say that because you understand it. You know that you financially or emotionally or whatever it is are not prepared to have a child, though you would physically be able. That's the same thing with the cell here at S. That's the puberty phase for the cell. Physically, it has the necessary genetic material to divide, but it doesn't have the necessary security. It hasn't accumulated. What I expressed to 7th hour yesterday was a college degree, a house, and a spouse. Those are the things you accumulate before you start a family. The cell hasn't done that yet. It does that during G2 right here.
That scared me. Oh crap, that was downloading.
So here is where it's going to accumulate all of the necessary proteins, all of the necessary enzymes, all of the necessary organelles. That's all going to happen. And once it does that in G2, then we're prepared for division. Now, as you think about it, human beings don't spend the vast majority of their lives reproducing, right? It's actually like a very tiny section of the rest of their life. And when we consider that, it's the same thing for a cell. The cell actually spends most of its time doing the everything else. And only a tiny fraction of it is spent actively dividing. That's why in this circle, which is why another reason I prefer a circle for a cycle, it kind of gives us an idea of that if we interpret the circle as a pie chart, right? Because mitosis, division, is such a tiny piece of the whole 100%. Makes sense. Now, mitosis itself, we do on a whole separate day from the rest of the cell cycle because it has a lot of parts. And what I don't like about all of the individual pictures is it makes it seem like it's one single, like one snapshot event. Keep in mind that all of these events actually occur in motion. It's not individual pictures. It's one fluid event like a video rather than pictures, which is why pictures I think are misleading. Oops. Skip. Aha, there we go. Figured it out. This is the exact image that you just looked at from your textbook. And the phases of mitosis that you are going to need to know are prophase, this first one, metaphase, anaphase, and telophase. As we look at those, you'll notice that there's a pro-metaphase in there, right? You're like, but hey, what about that one? Every step of mitosis has like a pro version of it in between it and the next one. So you have prophase, pro-metaphase, metaphase, then there's a pro-anaphase stuck in there as well. It's just the way that scientists label the steps in between each particular section of the cycle. So The one thing that I don't like here is it doesn't include what interphase looks like. A cell during interphase has a visible nucleus, so you can see its nucleus, you can see its nucleolus. So interphase has a nucleus and a nucleolus that are visible. You cannot see chromosomes. In interphase, there are no visible chromosomes. So in interphase, there are no visible chromosomes. You can see a nucleus, you can see a nucleolus, but you cannot see chromosomes.
Which one's interface?
Interphase isn't drawn on here. That's why I said I don't like this image, because it doesn't include that.
So should we write that?
You can include interphase. Remember, interphase is just G1S and G2. And when a cell is in G1S or G2, it's going about its life business. There are no visible chromosomes. There's nothing about it that indicates it's dividing. It just looks like a cell. Nothing special, nothing unique, just a cell. As it prepares for mitosis, which is the division of the contents of the nucleus, everything in the nucleus needs to become organized so it gets divided perfectly evenly. The first step of that organization is for chromosomes to start to condense. So the chromatin that we saw yesterday inside of our cells is going to spiral and wrap around those proteins, those histones, and become those X shapes. That's what happens during prophase. So chromatin condenses into chromosomes during prophase.
Chromatin what?
Condenses into chromosomes during S phase. I'm sorry, during prophase. Holy smokes, where am I? Hey, brain. A couple of other things happen during prophase that your book doesn't list. You'll notice in the picture that you drew, you have little, in the picture from the book, they're yellow, little yellow centrioles. Those centrioles used to be lined up next to each other. So these things right here. That one. Oops. Ah! Broke. Uh-oh. So those little yellow centrioles, they used to be directly right next to each other. During prophase, they start to migrate to opposite sides of the cell, so they move apart. Also during prophase, you'll notice these little black lines right there. Those are spindle fibers. Those begin to form from the centrioles. So spindle fibers begin to form from the centrioles. What your textbook lists as prometaphase is sometimes also lumped into prophase. So it's a phase between the initial parts of prophase. Oh, prophase, the nuclear envelope dissolves. So there's not going to be a visible nucleus anymore. It disappears. That's prophase. Nuclear envelope dissolves.
What's that?
Prophase.
I mean, like, what's the...
Nuclear envelope.
Yeah.
It's the outside of the nucleus. So the membrane that used to keep the nucleus separate from the rest of the cell is going to dissolve and fall apart.
So there's still a nucleus.
There's still a region of nuclear material of all that DNA, but there's not going to be a separate border anymore. Which you can see here because this cell, here's the cell membrane. This cell doesn't have a nucleus anymore. It has nuclear material, all that green stuff and that purple stuff, but there's no nucleus.
What were those yellow things called?
Centrioles. C-E-N-T-R-I-O-L-E-S.
And you said the black lines are the
Spindle fibers.
And what are they doing?
So they're starting to form here. So they're brand new. They're made of microtubules and microfilaments. And they're going to start to form because they have a very important job coming up in metaphase. during prophase or prometaphase, you'll notice those spindle fibers in this picture are attaching to the centromere of the chromosome. Remember yesterday on that picture of a chromosome I drew this little thing that I said stapled the two sister chromatids together? That's where the spindle fiber is going to attach, right onto that staple. So right onto the centromere. And now, the centrioles are on opposite sides of the cell. So you'll see here there's one on the left, one on the right. We call those the poles of the cell. If you turn your head, it looks more like a globe with the North Pole, South Pole. So those are the poles of the cell. We have our sister chromatids held together by our centromeres. We have chromosomes, spindle fibers, and no nucleus, right? There's no nuclear envelope that's visible here. The next phase that starts in mitosis is called metaphase, and it's in essentially the middle. Meta always made me think of the middle. And the reason that it makes me think of the middle, besides being sandwiched in the middle of mitosis, is because all of the chromosomes are going to align down the middle of the cell. So they all stack nice and neatly right in the middle. And the reason that they do that is because spindle fibers have attached to their center. And there's one on the left and one on the right. And if the centrioles are on opposite poles of the cell and pulling equally hard on those chromosomes, then they're going to be right in the middle. Think of it like a game of tug of war. You have that little rope or that little bow in the middle of the rope during tug of war. You know the one I'm talking about? When's the last time you guys played tug of war? You saw tug of war in October, right? So, Spirit Games Tug of War, when it started, you saw the ribbon, flag, bow, whatever it is in the center of the rope, directly in the middle. And when both sides are pulling equally as hard, that stays in the middle. If one side pulls harder, where does it go? To that side. What if I cut where that bow is? It's going to split evenly, right? They're going to fly to opposite sides of the cell, the gym. That's what's going to happen in anaphase. Once metaphase gets all of those aligned at the equator, the centromere is going to fall apart. And then sister chromatids get pulled to opposite sides of the cell, just because the spindle fiber acts like that rope. Sometimes I compare it to like a fishing line and a reel. So the spindle fiber is the line, and if you reel it in, it's going to pull whatever's on the other end, ideally a fish, if you're doing it properly. It's going to pull it in. If you're not doing it properly, it's probably an empty hook. It's true. I haven't been fishing in a while. We should try that sometime soon.
We should all go and say it's fun.
Fishing is fun.
It's like so, like, if you hit like, cause there's
Until
like,
a fish bites you.
no, it's like, whenever I go, like, it's always like in this, like, really, like, close to, like, the edge of, like, a swamp. And it's like, there's so many bugs that go by.
You want to know what my problem is? I insist on fishing in a boat on a lake.
Yeah.
So that way, if you go out far enough, you're pretty bug-free. But really, last time I went fishing, my dad said I was treating it more like an opportunity to have a snack and sit and talk. Which is like, you know how people are always like, you have to be quiet when you're fishing. Fish won't bite if you're talking. That is a lie.
Fish can't hear you.
That is a lie.
Yeah,
It is
that is.
just to get people to be quiet. My dad used to tell me that all the time when I was a kid. The fish aren't going to bite. When I realized that he was being a maker-upper, I talked twice as much the next fishing trip.
I'm not sure my dad stopped using that. Just told me to be quiet.
I probably would have cried. I was a sensitive kid. Like, if you would have looked at me and been like, be quiet, I would have been like...
How old were you?
No, I would have cried up until I turned, like, 31. So... some things you don't grow out of. Anaphase we covered, right? Telophase. you'll notice your textbook makes it look like this little purpley thing. That purple thing is a new nuclear envelope forming. So now we're going to have 2 nuclei, which was the point of this process. We wanted to divide the nuclear material in half. So we have 2 nuclei. It's also convenient then because this is when cytokinesis occurs. Now, well, I should say this is the start of cytokinesis. Remember how I said cytokinesis isn't part of mitosis, but it's always drawn at mitosis because it occurs simultaneously to the last phase of mitosis? A lot of people think that this indentation and this indentation here, we call them furrows, they think that those furrows are part of telophase. They're not. Those furrows are the beginning of cytokinesis. What happens is, during cytokinesis, the cell membrane is going to pinch in from the sides. And eventually, it's going to keep getting closer and closer and closer together until these 2 sides right here meet and make 2 individual smaller cells. You have 2 identical daughter cells, and cytokinesis is complete. Now, this furrow process is only in animal cells. Plant cells can't do that. The reason plant cells can't do that is they have this crazy thing called a cell wall. It's rigid and not flexible, so it can't bend in. So when a plant divides in half, instead of making a furrow, it's going to make a cell plate down the center of the cell. The cell plate will start to form at cytokinesis. This is a furrow, F-U-R-R-O-W, sometimes also called a cleavage furrow, because a cleaver, right, means to cut. So this, and the intention of this furrow is to cut the cell in 2, so we call it a cleavage furrow. And furrow, of course, means indentation, right? Like you all can furrow your brow. Show me your best brow furrow.
What does that mean? Angry face?
Furrow your brow. Sometimes people furrow their brow when they're worried, upset, expressing consternation, confused.
I have a question. What's the definition of anaphase?
So anaphase is where the centromere dissolves. I've been getting the hiccups a lot lately. 1 second. I had to stop that because I started seeing spots. I don't know what's worse, passing out or having hiccups. Just gonna have to sit. That's true. When you pass out, you wouldn't pay attention to your hiccups. Yeah, there you go. So anaphase is when the centromere dissolves. And sister chromatids, these little arms of our chromosomes, get pulled to opposite sides of the cell.
So the little arm?
Mm-hmm. Now remember, we said the point was to make 2 identical daughter cells, right? And that means that both cells have to have a full complement of exactly the same of all of the DNA. But because of the way DNA packages up into chromosomes, it guaranteed that was going to happen. Because here, in this phase, remember this arm of this chromosome is identical to the one right next to it, right there. If the thing in the middle holding the 2 identical parts together falls apart and they get pulled to opposite sides, that guarantees each new cell has exactly the same DNA. It's brilliant. It's one of those things that you're like, how did this, I mean, it's complicated, but brilliant at the same time. Like, problem with the cartoony versions is they don't really look anything like what the real life version looks like. I like this image because it has the cartoony versions down on the bottom. I like this prophase because it shows the nuclear envelope dissolving. You can barely hardly see it, for which I apologize. Cytokinesis, but this is the more important thing, what it looks like in real cells. So let's do this.